Today, I'm going to discuss about the topic cells of the immune system. Lymphocytes or lymphoid cells. Lymphocytes occur in large numbers in the blood and lymph. As we all know, lymph is the colorless fluid in the lymphatic vessels that connects lymph nodes in the body to each other and to the bloodstream. You can see in the diagram, the pink dots represented are the lymph nodes, and connecting the lymph nodes you can see the lymphatic vessels. And you can see lymphocytes also in the lymphoid organs such as thymus, lymph nodes, spleen, etc. Thymus and spleen can also be seen in the picture. Now, lymphoid cells are subdivided into three categories, namely B lymphocytes, T lymphocytes, natural killer cells or NK cells. B lymphocytes. B lymphocytes or B cells mature in the bone marrow. B lymphocytes play a critical role in pathogen-specific immunity by producing antibodies. Pathogen-specific immunity means we know that antigens are usually found on the surface of the pathogens and they are unique to that particular pathogen. The immune system responds to the antigens by producing special proteins called antibodies that directly attack the pathogen. This is called pathogen-specific immunity. Now, when a naive B cell which has not previously encountered an antigen first encounters the antigen that matches its membrane-bound antibody, the binding of the antigen to that antibody causes the cell to divide rapidly. Its progeny differentiate into memory B cells and effector B cells called plasma cells. So that is when a naive B cell, that naive B cell which has not encountered any antigen before, for the first time when its membrane-bound antibody and the antigen that entered into the body, when it matches, the binding of the antigen and antibody occurs. This causes the cell to divide rapidly, producing progenies. And these progenies differentiate into two types of cells, memory B cells and plasma cells. Memory B cells have longer lifespan than naive cells and they continue to express the same membrane-bound antibody as their parent naive cell. While plasma cells do not express membrane-bound antibody. Instead, they produce an antibody in a form that can be secreted. Although the plasma cells live only for a few days, they secrete enormous amount of antibody during this time. So, memory B cells have longer lifespan and plasma cells have a short lifespan. But memory B cells, they will not produce antibodies like plasma cells. Now, the secreted antibodies are the major effector molecules of humoral immunity. Humoral immunity produces antigen-specific antibodies. T lymphocytes. T lymphocytes also arise in the bone marrow and migrate to the thymus gland to mature. So earlier we learned that B lymphocytes mature in the bone marrow. Here T lymphocytes mature in the thymus gland. Like B lymphocytes, T cells have membrane receptors for antigen. T lymphocytes are involved in the regulation of immune response and cell-mediated immunity. B lymphocytes was responsible for humoral immunity, whereas T lymphocytes are responsible for cell-mediated immunity. That means it does not depend on the antibodies for its adaptive immune functions. It is mainly driven by mature T cells, macrophages, release of cytokines in response to an antigen. But in humoral immunity, we learned that they produces antigen-specific antibodies. Okay, it also helps B cells to produce antibody. T lymphocytes are classified into three types. They are T helper lymphocytes, cytotoxic T lymphocytes, natural killer T cells. T helper cells and cytotoxic T cells can be distinguished from one another by the presence of either CD4 or CD8 membrane glycoproteins on their surface. T cells displaying CD4 generally function as T helper cells, whereas those displaying CD8 function as cytotoxic T cells. T helper lymphocytes, adhesion molecules that binds to class II MHC molecules. Cytotoxic T lymphocytes, addition molecules that binds to class 1 MHC molecules. That means the main difference between class 1 MHC and class 2 MHC is that MHC class 1 molecules present antigens to cytotoxic T cells with CD8 receptors, whereas MHC class 2 molecules present antigens to helper T cells with CD4 receptors. Now, natural killer T cells. They are heterogeneous group of T cells that share properties of both T cells and NK cells. They recognize foreign lipids and glycolipids and constitute only 0.2% of all peripheral blood T cells. Natural killer cells. NK cells are large granular lymphocytes that kill tumor cells and virus-infected targets. NK cells constitute 5 to 10% of lymphocytes in human peripheral blood. NK cells does not have membrane receptors for antigen, but they have NK cell receptors for killer activation KARs and killer inhibitory receptors KIRs. The main function of NK cell is to kill self cells which contain viruses as well as some tumor cells. When NK cells bind to uninfected cells, NK receptors KIRs provide a negative signal to the NK cell, preventing it from killing the self cells. So we learned that the main function of NK cell is to kill virus infected self cells. But when it is bounded to uninfected cell, NK receptor KIR provide a negative signal to the NK cell and it prevents from killing the self cells because they are uninfected cells. The mechanism of killing by NK cell is mediated through the release of its granule contents, perforins and granzymes onto the surface of the infected cell. Perforins can punch a hole in the cell membrane, allowing the passage of granzymes. Granzymes are proteolytic enzymes into the cell and that induce apoptosis. Apoptosis means programmed cell death.